episode of the BioHub. Today, we're excited to host Tatiana Novobrantseva, a scientist and biotech leader who's been at the forefront of some of the most exciting breakthroughs in immunology. Tatiana is the CSO at NextPoint Therapeutics, where she's driving innovation in precision immuno-oncology and developing new therapies to harness the immune system in cancer. Tatiana's career spans leadership roles at companies like Moderna, Verseau Therapeutics, and Alnylam, and she's also co-founded Verseau Therapeutics. So join us today as we'll talk through Tatiana's journey, NextPoint's pipeline, and Tatiana's role in business development as a CSO. So Tatiana, thanks so much for coming on. How are you?
I'm doing well. Thank you very much for having me. It's really a pleasure to be here.
Yeah, it's a pleasure to have you on as well. So thanks so much for coming on. So look, let's start off with a little bit about your journey then. So you've had a career that spans across academia, pharma, biotech, and now cutting edge immunotherapy. Can you walk us through your journey into immunology and what's kept you excited about the field over the years?
It's a wonderful question, right? Because immunology has not always been a wonderful field to be in from the external perspective. But for me, it has always been really the centerpiece of how everything has evolved. I always like to tell the very beginning of the story, right? So I have hay fevers, and they were pretty violent. And there were absolutely no drugs, right? No antihistamines back then. So after very long winters in Moscow, right, when everyone was outside and enjoying the sunshine that finally was there, right, I was absolutely stuck inside, right, with my nose against the window, thinking, I mean, what the hell? Like, why? Why is this happening, right? So that was really a very personal, very simple, I guess, reason to start wondering how the immune system works at all, right? And we're really talking about school, right? So elementary school. And that's how it sort of eventually led me to become an immunologist, right? So I did my PhD at the University of Cologne with Klaus Rajewski in B-cell immunology, and really digging into what makes our immune system react, right? What is the enemy and how does it clear out that enemy? Becomes a very, very interesting question. And now, with a lot more that I know, a lot more that the field knows, right? It's really not a simple question, and we don't always have the answers, especially when designing drugs that we try to target very precise populations of immune cells to either attack cancer or to help fighting an infectious disease or try to revert fibrotic disease. I mean, any type of inflammatory consequences, right? It is still a complicated, not 100% answered question. But nevertheless, that's how my interest in immunology came together, right? And stayed for years, right? So obviously my PhD and postdoc were very basic, very fundamental B-cell immunology with a lot of genetic manipulations and making mice that express only one possible receptor specificity on B cells and seeing how those B cells actually behave if they fill the whole organism. Then I went into industry, and my first industry job was at Biogen, and that was involvement in a lot of inflammatory type projects. There were a couple projects that really concerned B-cell development, but we also studied a lot of inflammatory and fibrotic conditions and found super interesting insights there. And then I went to Alnylam, and Alnylam was first and foremost about making nucleic acids a drug, right? So siRNA as a new therapeutic modality. And back then, no one believed it could be a therapeutic modality, right? So being an immunologist there was an interesting proposition because it certainly brought a unique perspective, and we had beautiful projects on trying to deliver outside liver, but that was certainly before its time because it's only now becoming popular. And back then, I've heard many different opinions. So come on, focus on some real organ, like liver, right? That is there you know where to find it. What are those floating immune cells? And then actually immuno-oncology responses came, right, in the field. So CTLA-4 blocking antibodies, PD-1, PD-L1 blocking antibodies started to produce amazing, long-lasting responses in cancer patients. And that's when suddenly immunotherapy brought immunology to the forefront. And now we are really in a mini renaissance of immunology, because both immunotherapy and autoimmune research are really thriving. So it has been a long journey. I hope I explained how I got interested, but then there has been so much happening that it was impossible not to stay interested.
Yeah. Good. You've obviously been in both academia and in industry. What do you think the big differences and similarities are between an academic and industry approach to immunology?
Well, so it's a question really that many people ask, because everyone who starts in science starts in academia, right? There is no other way to start. You start with getting some education, right? It doesn't matter which degree you get. And then you either switch or stay, right? Or collaborate. And now actually, I think that with time, a lot of the boundaries blur, which is a wonderful thing. And a lot more people are involved in both or have been switching back and forth. But again, when I was graduating from my PhD program, it was still pretty frowned upon to go into industry research, because if you were a real scientist, right, you needed to stay in academia and sort of dig very, very deep into a particular discipline and set of questions. So there has always been a little bit of maybe higher meaning in academic research, right, which I certainly was under the spell of, right? And when I got a phone call asking me if I'm interested in interviewing for a position at Biogen, I actually said that I was not, right? Thank God I was convinced to go and interview. And when I came into the building and started talking to people who actually worked at Biogen, right, I realized they were absolutely phenomenal scientists with great publication records. But in addition to driving science and getting to the publication, they had a possibility to actually develop something that will change how we treat disease, and it will change people's lives, right? So for me, that was such an eye-opening experience, and I was absolutely naive to what industry is, especially in the US, because, I still think that there is a lot less biotech in Europe still today, even though the field has expanded and flourished, and it is absolutely amazing. But just by sheer volume, right, there's still more here. Obviously, there is huge competition now coming from Asia, in particular China and Singapore and Korea, right? So there is a lot happening everywhere. So this is all wonderful, but just having that. So I think that that's the main difference, really, right? That your project can contribute to making something that people use within your lifetime, rather than contributing to the fundamental knowledge, which is incredibly important, right? And without that, pretty much nothing would have been possible today, right? But might not contribute to anything that people can use. So this is not, I think that both are incredibly fulfilling careers. And at the end of the day, it's a personal choice. And one can be involved in both, which is wonderful. But that's for me maybe the only difference.
Yeah, very good. So you joined NextPoint as Chief Scientific Officer in 2024. What drew you to the company, and what made this opportunity stand out to you at this stage of your career?
So yes, I joined in January 24, right after JP Morgan conference was over. I didn't want to participate in JP Morgan conference 2 days after joining the company. So NextPoint, as usual, as with every opportunity in life, right? The important thing for me is science and people, not necessarily in that sequence, but those are the two driving things, right? So what is done at the company, right? What is it based on, and what the company is developing, and the team surrounding that. And the team, of course, is involving everyone, right? Founders, investors, the actual operational team. And NextPoint manages to combine all of that. It's super innovative science, great execution, phenomenal investor support, and absolutely wonderful founders. So it was just an amazing opportunity. So I was very, very lucky to have had that in front of me and for the ability to join.
Yeah, very good. So NextPoint's pipeline includes 2 clinical stage programs and 2 INDs on the horizon. Can you tell us more about these programs, what makes them different, and where you see them making the biggest impact?
So NextPoint is all around B7H7, which is also called HHLA2, and the 2 receptors to the molecule. And while being centered around that pathway, which is not found in rodents, so not found in mice and rats, but is present in primates, both non-human primates and humans. It's an incredibly innovative space because people didn't have the ability to study that in mice, so it always was very difficult to actually get to studying that because you had to be within human systems. And the 2 programs that are in the clinic are immunotherapeutic programs because B7H7, through those 2 receptors, one is KIR3DL3 and another one is TMIGD2, is either inhibiting or stimulating different immune populations and regulating the immune responses. So the first 2 programs are really immunotherapeutic approaches. The first one to inhibit KIR3DL3, which is an inhibitory receptor, if you like, an analog of PD-1, even though it's expressed in a completely non-redundant and independent setting and on different cells. But the second program is really inhibiting B7H7 on the tumor cells, but also by that inhibition, limiting the interaction with KIR3DL3, but opening up the interaction with TMAGD2, which is a co-stimulatory receptor. So trying to activate the immune, both approaches try to activate the immune system to attack cancer. Whereas the first program is what we call a pure antibody that has just the blocking ability as much as we can stay within one mechanism. The second antibody also has enhanced ADCC activity, so it can, through the Fc receptor, activate NK cells and macrophages to really kill the tumor directly, in addition to blocking the inhibitory mechanism through B7H7 and activating the activatory mechanism. So those are the 2 programs in the clinic, right? But then while working, and when the company was really started, it was an immunotherapeutic company that deals with an independent checkpoint, right? That is heavily present in PD-1 non-responsive populations of patients and has completely non-redundant regulation in the immune system. However, while working on this within the company, we realized that B7H7 can also be used as a tumor target because it is highly upregulated on a big array of human tumors. And it's only expressed really on one tissue, and this is gut epithelial cells, gut-associated epithelial cells in the normal tissue. So there is a lot to be said that we can actually target that molecule and cause direct tumor disruption and also potentially add immunotherapeutic effect to that destruction. So while destroying the tumor, we can stimulate the immune system to recognize what's being destroyed to keep the responses really long term. So that's the pipeline, and it's a pretty amazing combination within one pathway to be able to do both and potentially combine those in one molecule.
Yeah. And you've worked on both tumor targeted therapies and immune modulating strategies. From your perspective, what do you think the strengths and limitations are of each approach, and how do you think they can complement each other in cancer treatment?
So I just told you that we can actually combine them in one molecule potentially, but let's maybe take a step back and talk a little wider than just NextPoint approaches, because your question is just to compare tumor targeting versus immunomodulation. And then we'll circle back to NextPoint approaches, because I think it illustrates a very, very interesting approach and example of how they can be combined. Anyhow, so tumor targeting is very difficult because tumor is derived from normal tissue, and it's extremely difficult to find something which is super specific on the tumor but is never present anywhere else. But if we find something like that, then of course we can just directly destroy those cells by targeting a toxic payload, by targeting the immune system towards those cells to destroy them, right? So that becomes easy, but that distinction is very, very difficult. And people are using now very different approaches, right? Either identifying tumor-associated antigens, which has been probably 150 years, maybe more now, long effort. And it has produced some wonderful results, but not that many. And now people are looking at what can be even more specific. Can there be a peptide which is presented by MHC complexes on tumor cells that is only produced by those tumor cells? And there are some, right? They can be derived from rearranged genes within the tumor itself, right? From aberrantly expressed genes, maybe from what is called dark genome. So from non-coding regions, right? But that are activated in the tumor specifically. So those approaches are wonderful if you have something that is super direct, but it's very difficult to find something super specific, right? And the second limitation of the approach is that the more specific you get, typically you get to smaller and smaller patient population, basically already coming to very individualized care, which again is becoming possible because we are becoming better and better at analyzing whole genome, whole exome, whole metabolome, whatever we need to, right? Things, but they are expensive, and if you can have a wider approach, it's always good if it's specific enough, right? So that's on the tumor targeting side. On the immunotherapy side, you are really trying to use the immune system and its natural ability to recognize the tumor and to re-stimulate responses, because by the time the patient is diagnosed, the immune system is losing the battle, right? The tumor is growing, so the immune system cannot control it anymore. So immunotherapeutic approaches are wonderful in a sense that they can typically be applicable to very wide patient populations, and are generally very safe because you're trying to reactivate pre-existing immune responses and generate new ones, but they are really generated in the most natural way. Of course, it also has the drawback that if you activate too many immune cells or not specific enough to the tumor, they start destroying healthy tissue, and autoimmune type responses are probably the main side effects of immunotherapy, right? So we are dealing with narrower populations versus wider populations, right? In tumor targeting versus immunotherapy, we're dealing with different side effects because of different payloads we use. But ideally, right, we all would like to see cancer just go away. Meaning we want to destroy the tumor. And if the tumor burden is very, very large, you likely need something that directly destroys the tumor, even if it's used for a very short period of time. But then you need the immune system to clear out the remains and keep the tumor from coming back. So ideally, we would want to combine those approaches, right? And for that, we need sort of a good arsenal of tumor targeting approaches so that hopefully every patient would have something that targets his or her tumor. And then immunotherapy to really give the long-lasting responses that will keep the patient tumor-free, right? So, and coming back to NextPoint's programs, we have a chance. Of course, unfortunately, it will not work for every patient because the patients for which it will work, hopefully, will need to express B7H7 on the tumor, which is a large population, but not every cancer patient does. But if we manage that we can actually target the tumor with CD3 engager or an ADC, but then within that very same molecule, we can stimulate the immune system, enhance the recognition of dying cancer, and keep the long-term responses. So that's the hope, working with a unique molecule that can serve both as immunomodulator and a tumor target.
Yeah, very good. The immuno-oncology field has evolved rapidly in the last decade. Where do you think the field stands today, and what do you think the next frontier looks like in immuno-oncology?
This is an amazing question coming now, right, in mid-2025, because right now the immuno-oncology field is not the most popular, I would say, compared to several years ago, especially. But so if we go back some 10, 15 years ago, right, immuno-oncology, immunotherapy, tumor immunology, all were really unpopular, very niche, small disciplines, right? Very few people involved. And no one believed it could be done because immune system is having such broad mechanisms. You can do nothing to it that will be tumor destructive and safe at the same time. You're either not stimulating enough and not doing anything to the tumor, or everything will just blow up. Then CTLA-4 inhibition, and then on its heels, PD-1 inhibition came and started showing long-term responses in melanoma, which before that was a death sentence, right? Late-stage melanoma, basically people were told to just go and put their affairs in order, right? And suddenly we found immunotherapy that caused responses. Unfortunately, not in majority of patients, but the combination of CTLA-4 and PD-1 probably can do very meaningful things for 40-ish% of those patients, right? So that's what created this humongous popularity. Everything was immuno-oncology. We can do a lot more. Let's build on that momentum. Let's try to get to the patients who are not affected by this, right? The field has exploded. But then within that explosion, right, people were trying to combine everything with PD-1 inhibition or use molecules that look exceedingly similar to PD-1 and are expressed in very, very similar populations of immune cells compared to PD-1. So until today, there is no second PD-1, right, which is broadly applicable, reasonably safe, and effective. So people started saying, okay, is it immuno-oncology or is it just PD-1? And obviously, I strongly believe that this is immuno-oncology, and we just need to change the approach where we're looking. And there are a lot of approaches targeting different cells, targeting receptors that are independently regulated from what causes upregulation of PD-1 and PD-L1. So we will find something which is very broad. But right now, the the tumor targeting approaches, maybe with a little bit of immunomodulation, and T-cell engagers, which by the mere fact that they engage T-cells directly, they become immunotherapy because T-cells are part of the immune system. But in essence, they are tumor targeting approaches, right? Because we bring T-cells via tumor-associated antigen directly to the tumor cell. So they're really taking center stage. And as I just said within the last 5 minutes or so, right, that makes the immunotherapeutic approach is very narrow, right? Because you need a very good targeting agent, which is difficult to find, especially for wider populations. So I think that the field is wide open, and we're really waiting for the next thing. Maybe I will comment on one more relatively recent development. PD-1 or PD-L1 VEGF bispecifics have been all the rage for the last year or so, on the heels of clinical data. So seeing responses in people who failed every available therapy, including PD-1. The jury is still out there. We haven't yet seen the full data sets that lead to approvals. So we're still waiting. But that combination engages the VEGF pathway in addition to PD-1 pathway. And VEGF pathway is certainly touching a lot on innate immunity as well as blood vessel formation and tumor supply. So this is venturing outside of just T-cell reactivation or activation. So hopefully that will pan out, or at least that will pave a way for differentiated approaches beyond PD-1 in very, very different patient populations and in very different immunological mechanisms. So I really think that all types of bispecifics, as well as just monoclonal antibodies activating other than just T cells, so either multiple cell types at the same time or activating myeloid cells or activating NK cells, will bring something that will be wide, effective, and safe.
Yeah, good. So your role currently as CSO, you're not just leading science, but you're also involved in shaping partnerships and strategic direction as well. What's your role in business development at NextPoint, and how do you see science driving those conversations?
So yes, I am very, very actively involved in both internal strategy shaping as well as external conversations with potential partners and or investors. It's a fascinating asset to have in your possession, to be able to talk science to very, very wide array of people. And at least when I was going through formal education, I was never taught how to talk to people who are not trained in science, right? So we are all trained to present to fellow scientists and to people who are really super well versed in exactly that field that you are studying, which is necessary because, again, if you need to do something novel, at the very least, you need to be able to explain why it matters and why it's novel to people who have been studying similar things, right? On the other hand, when you're talking to potential partners and investors, it's a very different conversations. It's a different set of people with the interest to see where this program can take you, right? How can we solve an unmet clinical need? And if we do that well, is there a potential to also do well financially, if we're talking about investors in particular, right? But also for pharma partners, right? At the end of the day, every company is a business, so they need to be able to sell the drug, right? And get the profit that will continue driving innovation and generation of the novel drugs. So it's it's fascinating from that perspective, right? But also bringing science into those conversations, I think, is super important because I know by now that the deals are best made by people who are actual business professionals, right? Because they pay attention to that level of comfort with the overall story. But giving that scientific perspective into this, right, I think is super important, because then we don't overpromise and we keep things bound to what we can actually show under any circumstances, right, with any amount of funding, because some things we just wouldn't be able to do in a reasonable time frame or under reasonable budgets, right? So that's one of the roles, but also explaining how unique and differentiated each approach might be is a phenomenal conversation to have. So I really think that that's also a very important contribution from the scientific perspective to those conversations.
Yeah, good. And you've led scientific teams in both large multinational companies and nimble biotech startups. What do you think the major differences are in terms of leadership style and execution across the two different environments?
This is always a super interesting question. And I don't know there are humongous differences, really. So people interact with a certain number of individuals, regardless of the company size, right? So if you work in a super large company, most of your interactions are internal, or all of them are. If you work in a very, very tiny company, most of your interactions are external, and you work with CROs or collaborators, etc., right? But there are only so many interactions one can have in a day, and that creates your network and team and sort of processes you need to drive. And those interactions, I don't think they change too much based on where you are, right? Is it a larger company or smaller company? I guess the only thing that really changes is the number of approval levels, right? So in a company the size of NextPoint, around 30 people, right, it's all very clear, right? You can make decisions very fast, and you know that there is literally less than a handful of people whom you can assemble within a couple days, right? And the decision is made. In a large organization, that needs to go through different levels, right? And sort of to reach a decision, you need to go through multiple different conversations and then bring a united front to the final decision maker, right? But other than that, I don't know if it really changes that much. And I really enjoy staying involved in both scientific detail and the sort of strategic level conversation, because I think that really creates that necessary connection so that we stay real and true to what is possible.
Yeah, good. And last question, and I know wanted to give you a bit of a heads up on this, but if you were to have dinner with 3 people, past or present, who would they be and why? They don't necessarily need to be biotech innovators, although they can be if you like. But yeah, 3 people for dinner.
So thank you for asking this question. Thank you for warning me, right? Because it's always a super tricky thing to come up with 3 names. And first and foremost, that question really made me think about people whom I can go out for dinner with in real life. And I should really do that more, right? So thank you for that. I think that was a great sort of call for action, right? But for the answer to your question, I will stay hypothetical, because unfortunately I cannot meet any of those people anymore. But the 3 names I want to bring to the conversation today the following. So one would be Marie Skłodowska-Curie, right? The famous radioactivity researcher who had ginormous success in identifying novel isotopes, right? And sort of determining what radioactivity actually is. But it would be just phenomenally interesting to ask how she saw her life, right? Because what we know now, of course, is always colored with historical perspective. And we know how unique she was and what's the size of her contribution. But how did she feel this day to day? I think that would be just a very, very, very exciting conversation with really no end to it. So that's one, right? The second conversation I would love to have would be with Barbara McClintock. So she is really a geneticist studying maize mainly. And she has been a humongous innovator, right? She actually found a way to visualize chromosomes. And then she was the discoverer of transposons, among many other things. She was also a person who looked at what centrosome and telomere are doing. So many things that now really ended up in Biology 101 books. And from, again, historical accounts, I never had a chance to hear her talk or present. She was also just in love with what she was doing, and she could talk about how she sees those genes jumping and how that really influences the genetic material from one generation to the other, and how one can possibly change that path, right? So to hear her passion about this, right, and how she stayed focused on something that very few people at that time believed is even possible, right? Or many people believed that all her explanations were completely wrong for many years, right? So again, to hear that would be phenomenal. And one more name, right? Because you asked for three. So the last name would be William Cawley, which I think brings us back to immunotherapy. So he is believed to be the father of immunotherapy. And over 150, 160 years ago, he was treating his patients with bacterial extracts. And about 10% of them or so went on to completely resolve their tumors. Of course, the treatment he was doing would have been absolutely impossible in today's world because every bacterial extract preparation was different. No one could control the dose. No one could control the side effects. So it's not something that we could do today. But this is certainly something that got people thinking about immune system involvement and the fact that causing inflammation can actually pretty much cure tumors, even though I know that in the oncology field, no one likes to talk about cures because unfortunately recurrence is a real thing. But in practical terms, right, this is phenomenal tumor control and tumor undetected state. So talking to someone like that, right, with the knowledge of today and hearing him explain how he was explaining the effects, right, would be just amazing. And I think connecting through history, unfortunately, is impossible, but would be great for the development of the field.
Yeah, very good. Well, look, three terrific answers there. And I think you've given a really unique perspective of your role, the immunotherapy space, as well as NextPoint, who I'm really excited to follow as well. So, yeah, thanks so much for helping us out with this. It was a pleasure to have you on.
Thank you. Thank you. I really enjoyed the conversation. Thank you for inviting me again and for all the questions. It was a pleasure.